Hello, hi. I would like to continue share the week 6 lecture, which is focusing on single cell protein. Thank you for recording those video on 4 major bioproducts. Today, I would like to focus on single cell protein as a model bioproduct. This diagram shows the conventional bioprocessing from the upstream to downstream processing. Okay, so from the preparation of culture, as I mentioned in the previous course, that cell culture, for example, bacterium subtilis, will be stored at negative 80 degrees Celsius, usually in glycerol, to protect the cell from cell lysis once taken out from the deep freezer. If we store in water, the cell will lyse. It will abrupt and causing cell lysis. Okay. Then when coming out from negative 80, it will go through a series of dilutions and inoculation into the flask. Usually before the flask, it will be safer to culture it on the slanting gel in a small bottle. Once there are colonies growing on the gel, then only it will be inoculated into the flask. Then it will be the inoculum preparation to the real fermentation. This is typically around 200 liter. If the large one will be for 1000 liter for very large scale productions. Then the cell will be after the fermentation. Yeah, so anything from the cell culture to fermentation process, we consider it as an upstream processing. Okay, from the broth coming out from the fermenter, we consider anything here until at the end, the final product. This we consider it as a downstream processing. So cell is usually harvested using centrifugation or filtration. Then it will go through a series of purification. We call it low resolution first, followed by high resolution. So for low resolution, usually will involve precipitation to precipitate our protein from the liquid phase. Yeah? So let's say if this is referring to extracellular protein in the fermentation broth. Yeah? So in this process, centrifugation, we remove the cell as the side product. So our soluble product is in the liquid form. So the liquid form will go through precipitation so that we extract the product from the liquid. Then once we have the soluble product in the liquid phase, it will go through a series of high resolution purification steps. Typically, these involve chromatography, for example, HPLC. If this is, for example, we use viral particle as the host to produce vaccine, we have to inactivate the viral, then it will go through polishing, viral filtration, dye filtration. Then there will be formulation. Formulation means that we have to dispense the protein, which is the, for example, the vaccine, in certain stabilizing liquid called buffer or salt or protectants, some sort of preservative to keep the protein stable. Then it will go through lyophilizations, which is a freeze drying. Then quality control and packaging to check if there is any residue of endotoxin or exotoxin from the host cell. Finally, the pharmaceutical drugs will be marketed. Of course, in the quality control, after the quality control and packaging, before it goes to the market, it will go through clinical trial, pre-clinical trial plus stage 1, 2, and 3 clinical trials. Okay, so this is the overall process of bioprocessing if we are talking about biopharmaceutical drugs as the main product. Okay, as for other industries such as food technology, for example, for beer productions, the downstream processing will not be so complicated. Yeah, so we can say that the most complicated one goes to drugs, because it is for oral consumptions and to treat certain disease. So the quality control is very important. Okay, in the nutshell, this can be seen in this diagram. Just now I told you already, inoculum. Yeah, if the fermentation is very large, like 1,000 liter, it will go through from inoculum to a medium size, then to a large size. So this is upstream processing, USP, for medium preparation. And in order to ensure the large scale productions and the management, in order to the capacity may be limited by the size of the bioreactor. So there will be train A, B, and C in order to facilitate better mixing pattern, to make sure that the mixing uniformity is good in each of the bioreactor. If the reactor is too large, the mixing might not be homogeneous. Okay. And this is also to avoid any risk of contaminations. If we divide the batches of the preparation into 3 channels, this will better safeguard the continuous operations. Okay. After the fermentation is conducted, the cell is proliferates, the OD, the optical density goes up. The cell will increase in density. They will produce the drugs, the protein we want, as the intracellular or extracellular protein. Then firstly, this will go through centrifugation. Centrifugation to remove the cell. So presumably, if our product is soluble, the soluble portion will come out from centrifuge. Centrifuge might not remove all the cell. There may be still some residue of cell. So it is best to go through the microfiltration to separate all the cell particle from the product. Okay, then finally we have our pure liquid phase coming in here into the storage tank. To be further ascertain the purity of the product, it will go through ultrafiltration to separate molecules of different sizes. Further on, it will go through the chromatography, high resolution chromatography, to further separate protein A out from the rest of other junk protein. And then there will be, at the same time, there will be a place to prepare the buffer. Remember, the buffer is used to further purify the protein, because even from the chromatography, there may be still other junk protein that need to have buffer exchange. Buffer exchange to kind of like washing the protein to be much cleaner. This process, we use dye filtration. In the dye filtration, we need to have a buffer, to wash the protein. Then the purer protein produced will go through ion exchange. Ion exchange is also a kind of chromatography, which will separate the molecule based on positive or negative ion. Usually after the ion exchange filtration, chromatography, the protein will be quite pure and it will be stored finally in the storage tank, yeah, but still there may be still some residue of the virus particle which will go through virus filtration, and then further on hydrophobic interaction chromatography is considered as the polishing, because if you further separate out some of the other junk contaminant by hydrophobicity. Okay, again, it will go to the storage tank, another dye filtration to polish again, and finally sterile filtration, usually using filter of 0.2 micrometer, and the product will be produced. Yeah, you can see such a strict process which is required for the productions of a drug. Okay, so this is quite common for a pharmaceutical productions flow. Okay, of course, if we are talking about food technology, the process will be simpler. For example, there will be about 1 to 2% of the yeast growing in a fermenter. Upon completion of the fermentation, we rely on decantation. It's a kind of sedimentation process to remove the yeast cell. Of course, in order to, this is a yeast cell plus some liquid, yeah. Then it will go through centrifugation 1, 2, and 3. Yeah, centrifugation will further separate the yeast from the beer drinks. Okay. Then after 3 times of the centrifugation process, most of the yeast cell will be separated out. Okay, and often this will involve repetitive of centrifugation. Yeah, effluent, as you can see, the effluent will still be recycled, combined with the new income feed of the fermentation broth to continuously remove any residue. Okay, finally, the yeast cell is already quite pure. It becomes like a slurry, like half solid phase, with only very little water content. Still, it will go through filtrations, often under vacuum conditions, to produce a dry yeast biomass, which can be used as a single cell protein. Okay, and this is another common diagram which is used to produce single cell protein. In this case, is, if you can see here, the substrate is oil sludge. Oil sludge meaning that if there is any oil and plus some solid, we call it sludge, mixed with a lot of the water. Yeah, presumably in the sludge there will be organic nutrient, source of nitrogen, protein, carbohydrates, carbon source as a substrate for the production of the SCP. One of the best example of this oil sludge is the palm oil meal effluent. We call it POME, P-O-M-E, which still have good value of oil, the organic metal, which can serve as a carbon and nitrogen source to grow some single cell as a protein source. We call it as single cell protein. Okay. So what is single cell protein? At a glance, this almost cover every type of the microorganism from fungi to yeast, algae, and bacteria. To be exact, yeast is also known as Saccharomyces cerevisiae. For algae, 2 common products are known as chlorella or spirulina. Usually I buy from the market, it looks like this, and it will be pelleted and it become a good source of protein and vitamins. And this one, I guess you can know, you can guess that this is yeast, yeah, yeast, which is also can be used as an animal feed as well as nutrient supplement for human. As you can see, SCP defined as the microbial cell or total protein extracted from pure microbial cell and can be used as a protein supplement. As you can see, this so-called protein source, it can exist either as an entire microbial cell, which is high in protein content. It can be as high as 70%, or sometimes the protein will be extracted, but this will cause higher cost. For SCPE, if it is for animal feed grade, the production hygiene level and also the process will not be as strict as the one for human. Yeah, so for the one for food grade for human, the entire processing cost will be at least 10 times more expensive. Okay, so these are some examples of the microorganism strains belonging to fungi. As long as you see Aspergillus, Rhizopus, these are the fungi. It looks like this, yeah. And for yeast, yeah, for yeast, Saccharomyces and also Candida species, it looks like this, yeah, budding. And for algae, it looks like this under the microscope. For example, chlorella or spirulina. And bacterium, the most common one, if you can see this rod light, we know that this is a Bacillus species, Lactobacillus as well. Other types like Pseudomonas is also quite common. Why is SCP so important? It has advantages of rapid succession of generation, as we know. Okay, before that, you look at this, right? It looks, if you are perhaps vegetarian or know anyone which are vegetarian, you can see this kind of meat-like product, which are produced from a kind of single-cell protein called mycoprotein. Yeah, this can be produced by fungi. They are high in protein content. You see, even up to 85% by dry weight. If you want to modify the texture and so on, it can be easily GMO, genetically modified. However, in many countries, especially in the European country, there's a very strict rules on genetically modified. Okay, and then the production of this kind of single cell protein using microorganism is not depending on the climate, not like we are rearing cow, sheep, and so on. During the winter time, it might be shortage of the green grass to continue rearing, so there is some limitation in the cold season. So just now I mentioned that the advantages, including the good nutrition, simple to grow. In some cases, even we can grow using carbon dioxide as a carbon source, or any bio waste or lignocellulosic waste, such as cellulose as a substrate. Yeah? But this will depend on the organism. Okay. But however, for the disadvantage is that some of this cell culture will contain some toxic contaminant from the host cell. Okay, for example, for bacterium, it also has the cell walls and also the lipid level that will cause indigestion or allergy reaction for human or animal consumption. So here are the summaries of the advantages and disadvantages when we compare across 4 major microorganisms. Yeah, for example, for yeast, it has lower nucleic acid content as compared to bacterium. Yeah, bacterium tends to have higher nucleic acid content. And also yeast, human being and also animal are more familiar with yeast over many decades, that it has been used as a nutrition supplement in the feed or as a health supplement. So the acceptability level is high. However, it can also cause some digestibility in certain consumer. Okay, for algae, it's easy to grow. The harvest, the yield is high. Yeah, but the disadvantages is that it also has the cellulosic cell wall, which is difficult to, not to say too difficult, a little bit more difficult to digest as compared to, sorry, excuse me, other strain. So for bacterium, as you can see, one of the major concern is a high nucleic acid content that will cause toxicity or allergic reactions upon consumption. Yeah, okay. So take some time to digest that. So as I mentioned just now, one of these SCP called mycoprotein. Yeah, originally it was grown using this microbe called Fusarium graminearum. Okay? Using molasses is a kind of brown sugar, or we can also use glucose to grow. And using ammonia and H3 as a protein or nitrogen source, and also to control the pH to be within 6 to 7. At the end, the product will go through thermal treatment to reduce the RNA, yeah, which is the nucleic acid content. And finally, the mycelium is a kind of mycelium, like a hairy, having the hairy structures, will be separated by vacuum filtration. The main features of this mycelium produce is due to their food texture that is mimicking meat. Okay, you know, for example, McDonald's in Singapore, they have this kind of microprotein based burger, which is produced from this kind of biotech using microorganism. Okay, at the same time, okay, microprotein actually it belongs to mushroom. Yeah, it is rich in many type of mushroom, but this is the only mushroom-based protein that has been approved for human consumptions. I have found this very useful PowerPoint from the website. You can click to go to the website. However, I have also downloaded some of the interesting information, yeah, as our course content. This is the commercial productions of SCP from hydrocarbon. Yeah, you can see just now we talked about the substrate can be from palm oil, effluent sludge, or from cellulosic waste, or can be from molasses and glucose. At the same time, from the in the early years, when it was first founded, using SCP to grow more protein for for animal or for human consumption, it was because after the World War, there was a food security problem. So this was one of the way to supplement the diet with high protein. Yeah. So in the old time, the substrate rely on hydrocarbons, which is from the distillation and cracking of the petrochemical to for the hydrocarbon, for example, methanol, ethanol, through the distillation process, the for example, methanol can be used as a substrate. Yeah? So it will be much cheaper and easily available at a large quantity. After the purification through distillation, it will be used as a substrate for specific type of microorganism that can they will grow well and become to propagate the biomass yield as a source of protein. Okay, so in fermentation, we need water, microorganism, and also mineral salts, a part of the hydrocarbon as carbon source. Yeah, and then there should also be air spudging, followed by separation or concentration. This is a liquid solid separation. For example, centrifugation, decanter, and also filtration. Then the SCP cell will be dried using, for example, vacuum dryer. And finally, we will have the protein product, which in the old time, mostly are used for animal feed. However, if some of the if we use a more strict hygiene requirement, it can also be used for human consumption. But for this flow, this one human food means that the animal is food for the human. Okay, so this is how the cycle for the protein. So at a glance, from the okay, when we talk about single cell protein, right, it is not referring to a pure protein. Generally, it refers to the whole cell of the four major microbes that I mentioned just now: bacteria, yeast, filamentous fungi, or algae. Okay, because this whole cell will contain carbohydrate, lipid, nucleic acid, mineral salt, and vitamins. Okay. However, our intentions is to maximize the protein content. Often, it was also manipulated using genetic engineering to enhance the protein content. In the old at a very large scale, as I mentioned, alcohol or any N-alkane, molasses, sulfate, liquor, and whey. Whey is a byproduct of cheese industry. Are often used to grow SCP. Okay, so you can see the summary of protein content, where bacteria recorded the highest protein content. However, it also suffer of high nucleic acid content, which is not good, because we need to put more effort to reduce the nucleic acid content. Okay. If we compare to soybean, which only contain 40% of protein, obviously this kind of a single cell protein will be a better source of protein. So this is a very good summary diagram which compare across the cultivation technology for these four major microbes in terms of growth rate, substrate selections. For example, bacteria, it can use a very wide range of the substrate, because some bacteria can even grow under anaerobic conditions or using CO2 as a carbon source. Yeah. Then the pH range for algae, it can be grown using a pond system, but for the rest, the best is using the bioreactor under sterile conditions. In terms of the contamination risk, algae, because due to the pond system, because it's an open system, so it will be subjected to higher risk of contamination. Okay. Then another concern is about sulfur containing amino acid. Yeah? So usually any protein that is of higher complexity and higher value will need sulfur as the main element in the amino acid. So for bacteria and fungi, since they are simpler cell as compared to algae and filamentous fungi, their content of these S containing amino acids are quite low. So this is a disadvantageous for bacteria and fungi. And however, in terms of nucleic acid, algae doesn't have any problem of nucleic acid. For example, the RNA. For the rest of the microorganism will require this process. Yeah. So also bacteria will suffer the problem of endotoxin, especially from gram-negative bacteria. It has a high level of endotoxin from the intracellular compartment. As for the filamentous fungi, which has a hairy structure, microtoxins will also exist. So this will need extra purification to ensure. For example, just now you saw so many stages of the chromatography purification, hydrophobic, ion exchange. Yeah. So their objective is also to remove all these endotoxin as much as possible, which will cause toxic effect to human being or animal or allergic reactions. And I have added, for example, many short sentences in red color at the bottom or top on many slides, which is to help you to imagine what kind of the questions that will come out in the test or final exam. Yeah? For example, the question will be discuss and explain the pros and cons of different microbial strains for producing SCP. So you can draw this diagram and elaborate. For example, they are in terms of growth rate 1, 2, 3, 4, 5, 6, 7. Yeah? 7 parameters that you can compare. Then so in the exam, if you can remember 3 or 4, that will be good enough. Okay, these are the example of the spirulina available in the market. Even if for human consumption, usually they will tablet it. Yeah, for animal feed, it will be just like in the powder form. Yeah, so this is mostly for human consumption. Then another example of the question will be for you to provide 2 species and the corresponding substrate used to produce the SCP. For example, if you like the lactobacillus species here, you can use glucose, amylose, or maltose. Yeah, of course, this is not definitely strictly like that, but these are compound from whatever that has been implemented in the industry. So far, these are what has already happened. Yeah. Okay. For example, for fungi, it is best to be to consume maltose and glucose as a substrate. Of course, you may not be able to remember everything, but you just try to pick, for example, for yeast, the substrate can be lactose, pentose, and maltose. Okay. Further on, as I mentioned just now, hydrocarbon, CO2, methane, methanol, ethanol, glucose, inulin, molasses waste. These are the common substrate that has been used in the industry, and these are the microorganisms involved. Yeah, so as you can see, quite easy to remember. For algae, it can use CO2 as the carbon substrate. Yeah. Then the question might be asking you about elaborating the pro and con of selecting petrochemical based substrate as compared to the bio waste as a substrate. Yeah, for petrochemical, the example is like methane, methanol, and alkane. For bio waste, this will example molasses is actually a side product from the sugar refinery, and whey is a bio waste from cheese. Yeah, inulin is a kind of polyfluctan. I remember it as a prebiotics also. Yeah, inulin is a pre. Then glucose can be also considered as a bio waste from the starch hydrolysis process. Okay, so you can see the range of the microorganism there here. Okay, and then I will not read all the content. I believe you can try to go through the scope. And the most important thing is to look at the red color, elaborate the pro and con of producing SAP using microbial versus the conventional process. So when we talk about conventional process, meaning conventional plant and animal protein sources. So for example, if we compare to eating meat as a protein source, what would be the advantages given by SCP? Yeah? For example, we can say in SCP, the protein can be as high as 30 to 80% on a dry basis, and the rapid growth. Yeah, all this, some of the content has been mentioned just now. And for growing SCP, we need a smaller area as compared to rearing a cow and sheep, that you need a much larger farmland. And most importantly, the production is independent of seasonal and climate variation, as I have also explained just now. Okay, and the quality produced is also quite consistent. Okay, I don't know where to put this one. All right, and further on, in terms of the, so this is replying the same question just now, yeah, the pro and con. So this will be the con. For example, the disadvantages of SCP. Okay, especially bacterial cells possess high nucleic acid content, which may prove detrimental to human being by increasing the uric acid level in blood. So this is saying that, you know, many male I know suffer of a gout problem due to high uric acid in the blood. Yeah, so this can be caused by high nucleic acid content in our food chain. Okay, so this the additional step to overcome this problem, to remove the nucleic acid by extraction, solvent extraction method, or thermal treatment. This will cause more extra cost. Okay, and then the question may be asking you to describe the 3 major phases for microbial fermentations. Although I know just now I talked only on 2 phases, right? The upstream processing and the downstream processing. Okay, to be exact, the upstream processing we can also divide into 2 phases. Yeah, the first one is medium preparation. So you must be able to elaborate what is the first stage, first step for fermentation in the upstream processing. So for example, it is to prepare the medium. How do you select the substrate? If your substrate is a complex polymerase substrate, that you need hydrolysis first before you can produce a simple sugar. And then you also have to choose the right choice for the nitrogen source, phosphorus, and other essential nutrients. Yeah, so this you need to elaborate. For example, just now we have already seen different microbes will prefer different substrates. For example, either glucose or maltose and so on. And then it is the core process of fermentation. Yeah, which has to be cleaned in place to prepare the aseptical conditions. How do we control all the parameters to make sure they grow at the maximum rate? I think in the previous lectures, I talked a lot about the parameter, for example, temperature control, pH control, and the foaming control and so on. And also I compared the SSF, solid stage fermentation, with the submerged fermentation. Yeah, okay. So you have to elaborate, especially on the fermentation process itself. Yeah, and this one is only for general knowledge. Yeah? There are at least 5 major processes that has been established to produce SCP, and they all will rely on different carbon source, microbe, or for different product quality checking. Okay. Just now, following the medium preparation, fermentation, and lastly is the separation and downstream processing, where the cells have to be separated from the medium using filtration or centrifugation. The spent medium, which is the liquid, we call it also as a supernatant, the liquid form. Liquid may be processed in order to reduce level of nucleic acid, thermal, for example, using thermal shock, yeah, to inactivate cellular proteases. Okay, so this kind of protease is a kind of enzyme naturally occurring in the bacterium, but this kind of enzyme may also cause toxicity and allergy for the human. Okay, so sorry, the spent medium most likely will contain the protein that we wanted to have. Yeah, for example, if the solid is a biomass, that is a okay. When we look at SCP, actually mainly we want the cell. We want the cell, but sometimes in the process of producing the SCP, there are also some valuable soluble product happening occur in the spent medium. Yeah, which they may also contain the residue of nucleic acid, which will need thermal shock for reducing to denature the nucleic acid. Okay, and often further purification involve solvent wash and dehydration or dewatering until we will get the dry biomass as an SCP. So coming back to this diagram that I have shown you just now, the possible questions in the test will be, you will be given this diagram, but you have to elaborate, explain how the upstream processes when involving hydrocarbon source as a substrate, and what are the rest of the process, fermentation to downstream processing, to produce high yield of SCP using bacterium, fungi, you know. And in the exam, of course, I will further identify or let you choose whatever bacterium or fungi that, or any specific SCP, for example, microalgae, spirulina that you want to produce. Okay, so you have to elaborate the upstream processes on how to safeguard a high yield. Okay, this also overlap with the point just now that how do you control the fermenter? How do you choose any equipment to separate the solid and liquid in order to get a high yield? At the same time, you have to propose a choice and justify the selection of the equipment used to concentrate. Concentrate mean here, concentrate or dry and package the SCP. Okay. I have given some example here. Yeah. For example, this is the further questions, elaborations? Yeah, okay. Based on this diagram, explain the process to produce vitamin B from a microbe. So you have to name the microbe which are used to produce vitamin B, for example, and you have to propose or explain why you choose certain substrate. And you explain the fermenter, meaning that the parameter control. And you have to describe, for example, in this process using oil sludge, inoculum. So when you explain the fermenter, you have to say in the fermenter, for example, for 10 liter, it needs the agitator to mix the culture to be homogeneous. It may have the water jacket to control the temperature at the optimum temperature, for example, 37 degrees Celsius. And then it needs to have an air spudger, usually at the bottom of the fermenter to provide oxygens or maybe nitrogen if it is for anaerobic conditions. Yeah, it depends on the microbe you choose. Then pH control, temperature control. And then you will have the oil. In this process, the main goal you have to explain that is to produce the bacteria of desire, the desired microbe that you have chosen as the biomass or the SCP. And since your substrate is oil sludge, you may also have the residue of oil in the water. When coming out into the this process, so this is upstream, and then here is downstream. For example, you, in order to, a part of produce the biomass as a SCP animal feed, finally, you may also have a liquid portion which contain the value added product, for example, vitamin B. Okay, so in this process, in order to extract as much as the vitamin B from the cell, you need the solvent extraction. Yeah, so in certain case, the solvent extraction is also needed to deactivate the nucleic acid. Okay, so this will depend on what kind of microbe and substrate that you have chosen. That a typical process for SCP production is, it looks like this. Okay, and then at the same time, most likely the solvent and the oil will be on the upper level. It will go for distillation for recovery, solvent recovery, so that some of the oil can be recycled back to the fermenter and the solvent can be recycled into here, solvent injection unit. Yeah, so this is a solvent and oil recovery process. And after that, going through the extraction in order to extract as much vitamin B as possible. The vitamin B will be soluble in the water. At the same time, the biomass will be drier, meaning using spray dryer to produce a dry powder form of the biomass as the animal feed. So this biomass actually is an SCP. And then in the centrifugation is to separate the solid biomass from the liquid, which have the value-added product. Okay, so this is one of the example of the entire process that as a master student, you should be able to elaborate the process like this. Okay, in order for you to explain better about product purification, example of unit operation. Unit operation means equipment. Yeah, in chemical engineering or biochemical engineering, we refer unit operation as the equipment for solid liquid separation. Okay, so there are four major downstream processing. The first one I like to share is decanter centrifuge. If you click, there is a URL hyperlink that you can go to the website. And better still, they have a video to see, to show how this sedimentation tank works. Okay, so this is one of the concentration equipment. Typically, for any feed that is high in solid content, for example, wastewater having the sludge, like a mud like that. You want to remove the sludge from the wastewater, you can use sedimentation tank. Especially we call it decanter centrifuge. Yeah. So if we use just a normal decanter where the sludge just sediment under gravity force is called 1G, yeah? Then we call it as a normal or conventional decanter. However, this one is known as decanter scroll. Is operated at a much higher forces, 3000 G, yeah, 3000 G, so that a kind of centrifugal force when the screw is turning, the solid will be concentrated at the outer, at the bowl, and discharged as a solid here, and the liquid will be pushed towards this right hand side and discharged through another side. Okay, so in this way, not needing very high centrifugation force, we are able to separate this solid discharge. Yeah? So say the percent, when I say high solid content, this can refer to 10%, above 10%, 20% of the solid content from the liquid. Yeah? So this is quite an useful method. Yeah? So you can read through, you can also watch the video. Yeah. If you click here, you can see the YouTube video showing the operation. Okay. The second one is called centrifuge. I hope you have seen a centrifuge. This is a industrial scale. The most common one is called D-stack centrifuge. Sometimes, generally, we can also call it as a separator because it actually refer to liquid, solid liquid separator. Okay. It looks like you have a feed, but okay, what is the differences of centrifuge as compared to decanter? For decanter, as I mentioned, it is for high solid content. Yeah, and you can see the mud. Solid content, but for separating of microorganism, algae, yeast, and bacteria, we consider this microorganism as ultra fine solid content. Then we will need centrifuge. Why? Because it needs to be operated at a much higher gravity force, represented by this unit called G. Just now, remember what was the force for decanter? Only 3000, right? So for centrifugation, it must be above 12,000 G, sometimes even go up to 30,000. Okay, and then it can be used to harvest algae, mainly to remove algae from the pond water, for example, and for beer fermentation, we call it beer clarification. We can use it to remove yeast, yeah, as a side product from the beer, which is the liquid phase, which is the product for sale. Then also protein extraction. Yeah, if we want to remove the protein in the liquid phase from the cell, we can also use centrifugation. Okay, so mainly there are two types. One is called two-phase, another is three-phase. As the name implies, yeah, two-phase means that we just remove the liquid from the fine solid. Yeah. However, for three-phase, which is spun at a much higher centrifugal forces, which will separate even liquid phases of different density, and this, of course, must simultaneously remove the solid. Okay, there are two interesting video that you can watch. One is two-phase, another is three-phase. I strongly encourage you to watch the YouTube. Okay. Number 3 is a filter press. This is the third unit operation used for solid liquid separation. Mainly, this is used for dewatering. And if you remember just now, I mentioned after centrifugation, there may be still some residue of the yeast. In order to eliminate further, we might use a filter press to remove, but at the same time, it is also useful to separate or to dry, to remove water from high solid content. Yeah. So having said that, if we do not want to do centrifugation directly, we do filter press, which is also okay. But the problem of filter press is that it might cause fouling on the membrane. Yeah, okay, that's why we usually do the centrifugation first, followed by filtration. Yeah, so filter press is it also represent a concept of filtration. In the conventional filter press, you can see how it works here in this YouTube. Very interesting. Secondly, this one is by this German company called Floatweb. Bioprocessing also has an interesting YouTube that you can watch. This is mainly for producing fruit juice, to remove the pulp away, going through a series of the belt pressing process, and finally we will have the pomade coming out here, okay, and the fruit juice discharge at the other end. Okay, another very interesting belt filter press is for removing water from cassava residue, cassava ubi kayu, yeah, in Malay language. Okay, I strongly encourage you to watch also. And the fourth major equipment in the downstream processing is this so-called spray dryer. Yeah, when we want to finally dry our biomass or our single cell protein, we will use spray dryer. Yeah, thermal treatment. So we have a heater, and then there is a fan. There's a fan flowing in air or nitrogen source. Yeah, depending on the requirement. So going through the heater is a kind of heat exchanger at close to, usually it's around 80 or 90 degrees Celsius. Yeah. If the temperature is too high, it's very energy intensive. Then the drying gas will come in here and flow in homogeneous conditions. At the same time, the fermentation broth, remember just now, after the centrifugation, the biomass, the single cell protein, is still a little bit wet. So it will go into this storage tank and being pumped into and being pushed through a nozzle under a pressure of about 1 bar or 2 bar. Then it will spray. When it is pushed through the nozzle, it will spray. Yeah? Like we spray the aerosol like that. When it spray, mixed with the flying and the turbulent flow of the drying gas, it will become powder, yeah, being dried as a powder and going through the cyclone. Cyclone also have a swirling gases to collect the main powder fractions here. So this is to improve the drying and let it settle. But in the same time, they will also have some residual powder going through another filter, which the fine one will fly like an ash like that, and the larger particle will settle down in the cyclone. Okay, so the fine portion here and the coarse portion there, and then going through the fan, some of the gas will go through the condenser here. If there is any liquid or solvent, it will be condensed for further recycling. Okay, especially if the biomass is treated with any solvent. Remember just now there was a solvent extraction to reduce the nucleic acid. So there may be some residual solvent will go through here. And the air, the recycled air will be recycled and heated again, where the process will continue. Okay, so this is I have already explained 4 major downstream processing that you should be able to articulate. Okay, This is the last slide for this lecture. I look forward for your further questions. Yeah. So in a summary, today I have cover the general form of the bioprocessing from upstream to downstream, taking SCP as the model process, where I compare the advantages and disadvantages of 4 major microbia to produce an SCP, and I elaborate about the productions of SCP production, comparing the substrate selections, microbe selections. Yeah, this is a recap again. And finally, I share 4 major downstream processing units with you. Okay, so I hope this lecture will give you some idea on bioprocessing from upstream to downstream. Thank you for listening. Thank you.